The Part 4 notes will be on transport of substances through the phloem tissues of plants. Phloem sap moves by a process known as translocation. We have talked about translocation before. It means to move. So anytime you hear the word translocation, it means we're moving something. We use this term as one of the steps of protein synthesis, right? In translation of protein synthesis, we said the ribosome translocates. That meant that the ribosome moves down the messenger RNA strand to help translate it into amino acids. In this case, what we're moving by translocation is going to be the phloem sap. And that means that these dissolved substances are going to be moved throughout the phloem tissue. So again, translocation means to move. We are moving organic solutes throughout the plant. This means that it's going to be anything made directly by the plant. This would include sugars that were made in cellular respiration. It would include bigger sugars like sucrose that needs to be moved for cellular respiration, for use in cellular respiration. Amino acids that are going to be used to make a protein will be moved by translocation through the phloem sap. And then any other protein that's made by the plant, including hormones, would also get moved through the phloem sap by translocation. In this process, the molecules that are being transported can be transported in any direction. So you can't really use the term like up, down, side to side, because it really moves in all of those directions. It's very different from where water is always moving in the upward direction. This time, we're going to use the term molecules are moving from source to sink. So that means wherever things are going to be made, that's the source, they will get moved to the place where they need to be used or stored. That's the sink. So sucrose, for example, sugars made by the plant, they're going to be made in the leaves, and then they are going to be translocated towards a sink. And there's lots of things that can be a sink, but think about it. We need to get those sugars made in the leaves to various locations, including the roots, and the roots are down. So it wouldn't make sense for us to think that sugars can only be transported up. They can be transported in any direction, and they will always be transported from source to sink. Sources can become sinks, and sinks can become sources. Examples of sources are going to be like a mature leaf, because a mature leaf is going to do lots of photosynthesis and create a lot of sugars. However, it's not really using those sugars necessarily for itself anymore, because what a leaf typically is going to use the sugars for is for growth, and a mature leaf is already fully grown. So it doesn't need all the sugars that it's making for itself. Therefore, any excess sugars made by a mature leaf will be transported or translocated to a sink. Examples of sink are going to be any part of the plant that needs those sugars so that it can metabolize them to do cellular processes. For example, young growing leaves, they need more sugars, so they would be a good example of a sink. Flowers, when flowers are developing before they are mature, it takes energy to be able to grow a flower, and so that would be another example of a sink. Roots, we already said, need sugars. We already know that roots do a lot of cellular respiration, and they can't make any of their own sugars because they grow underground. So roots are going to be a good example of a sink. And then anything that's going to store excess food for later, for example, the root vegetables, right? So we have carrots, radishes, potatoes. All of that is like a big bundle of starch, a bunch of sugars that are growing underground, and we are storing those sugars for use for later in the plant. And so this is a good example of where a sink can become a source. So when we think about the life cycle of a carrot, The carrot is actually on a 2-year life cycle, and it spends the entire first year growing the carrot underground. And so we're constantly using those leaves that are sticking out of the ground to create the sugars and then translocate them to the sink, the carrot. If you were a farmer or a gardener, you would pull the carrot at the end of that first year and you would eat it. However, in nature, if a carrot was left to grow, in its second year of its life cycle, the carrot itself, the root vegetable itself that used to be a sink, will become a source, because the carrot will now send all those stored carbohydrates upward towards the developing flowers, which is now the sink. So again, this is a really good example of where our sink has now become a source. Translocation is going to be due to differences in pressure. The phloem sap is going to move from an area of higher pressure to an area of lower pressure. This high pressure environment will be created during the process of translocation. So we'll get into some of those details. Phloem loading is specifically when we load the sugars into the cell itself, the phloem cells itself, to be ready for transport. And this is what will help to create those pressure differences, loading a bunch of sugars into our phloem cells. This is going to be an active transport process to get all these sugars into the phloem cells to be ready for translocation. The sugar that will be used for transport will be sucrose. It's not typically glucose. This is because sucrose is a disaccharide and glucose is a monosaccharide that is the most commonly used monosaccharide in cellular respiration. So if we tried to transport glucose through the phloem sap, it would get easily metabolized along the way because it's so easy for the cells to just grab onto that glucose and use it in cellular respiration. So instead, we transport the sugars as a larger sugar molecule, sucrose, that will be able to be used for transport because it's not directly metabolized by the plant. It would have to be broken down into smaller sugars before used in cellular respiration. So sucrose is a better transport molecule compared to glucose. Glucose ultimately does get converted to sucrose. So remember, we talked about photosynthesis in a previous unit, and we said that the light reactions first make the energy molecules needed for the Calvin cycle. And then in the Calvin cycle, in the chloroplast, carbon dioxide is taken in, and that CO2, along with the energy from the light reactions, gets converted into a 3-carbon sugar specifically, and that 3-carbon sugar is called triose phosphate. Triose phosphate, we said at that point, would be converted into glucose. Glucose is a 6-carbon sugar, so we have our 2 3-carbon sugars that can be combined through enzymatic reactions to create glucose for the plant. However, those triose phosphates can be metabolized into other types of sugars. And so in this case, we'll be turning our triose phosphates into sucrose. Triose phosphate, after it's made in the chloroplast in the Calvin cycle, then gets exported into the cytoplasm of the mesophyll cell. Once it's there, it will go through a series of enzymatic reactions, which will convert multiple triose phosphates into sucrose. That sucrose is now ready for transport. So let's take a look at a picture on the next slide. You do not need to know the details of this slide, but what I want you to see is, yes, we talked about this in a previous unit. Here's our Calvin cycle, making triose phosphate, our 3-carbon molecule, that goes into the cytoplasm, that then goes through a series of enzymatic reactions, and ultimately we will make sucrose from it. So you should just see, yes, wow, that really is a lot of enzymatic reactions that are used to make the sucrose from the initial triose phosphate. So once we have the sucrose, it can be loaded in a couple of ways. Sucrose can travel through plasmodesmata, in which case if it does, that means it goes from cell to cell to cell. So from, for example, the photosynthetic cells that made the initial glucose and then converted it into sucrose, until ultimately it gets to a phloem cell. So that can be a companion cell or a sieve tube member. That's one way. So again, through plasmodesmata from cell to cell to cell. That's shown here with the blue line. We make the sugars here, and then we transport them over here to our phloem cells. Or it can travel through the cell walls, or apoplast. Remember, that's what that term is called. Sucrose can travel through the apoplast, or cell walls, between the cells of the mesophyll and get loaded that way. So here we have, you can see the red line. If you follow the red line, ultimately we have to get the sucrose out of the cell walls and load it into our phloem cells. If it's moving through the cell walls, the process is more complicated to load it. If it's moving through plasmodesmata, it's just a direct loading into those cells. So that process is pretty simple. So let's get into the details of phloem loading using sucrose that is moving through the cell wall. We're going to focus on this picture that is blown up over here. So when sucrose travels through the cell walls, here it is in the cell wall, we have to have a way to bring it into the cell. And so we're going to use our chemiosmosis by using co-transporters. So we talked about this earlier in a different set of notes where we have our proton pump embedded in the membrane. This is going to be used to pump hydrogen ions from low concentration to high concentration. Then we get our buildup of hydrogen ions happening here in the apoplast. Hydrogen ions are then going to come back down their concentration gradient by facilitated diffusion, but when they come back down, we're going to use this co-transporter. And this co-transporter, when it brings in a hydrogen ion, it will also co-transport a sucrose in. Sucrose is being transported against its concentration gradient, but we're not spending any additional energy to activate this molecule to bring the sucrose in. We already spent the energy with the ATP to activate the proton pump, and then we have our buildup here of our hydrogen ion concentration. That is potential energy. So this potential energy is where we get the chemiosmosis part of this that is going to be used to co-transport sucrose against its concentration gradient and bring it into a phloem cell. So sucrose here has now been loaded. So that's important. We've now loaded our sucrose, but this time we had to use the co-transporter and we had to actively load it in because it was stuck in the cell walls and we needed to get it into the actual cell itself. So this process can happen directly into a sieve tube member, which means it's been loaded, or into a companion cell, which also would mean it's been loaded because the companion cell will just feed the sieve tube member that sucrose through plasmodesmata. Remember, that's the job of the companion cell, is to transfer whatever is needed to the sieve tube member, since they are hollowed out and don't have anything in them except for space, really, to sort of transport things through translocation. So here's just another picture showing that. You don't need to be able to name these pathways, but you should have an understanding that in order to load sucrose from wherever the sucrose was made, so near the palisade parenchyma, for example, it can go two pathways. It can go a simple pathway where it just travels from cell to cell through plasmodesmata, and once it reaches a companion cell or a sieve tube member, it's been loaded. Or if sucrose is moving through the apoplast, the cell walls, that process is a little more complicated and does involve active transport. We would need to use a pump to pump out the hydrogen ions. We're going to get our buildup of hydrogen ion concentration here. Hydrogen ions will come back down their concentration gradient by facilitated diffusion, and with it, they will co-transport sucrose in using the same molecule. Sucrose is moving against its concentration gradient, and we didn't spend any additional energy. We're using this chemiosmosis part right here to load the sucrose in. So sucrose has now been loaded. And remember, it can be this right here, can be in the companion cell, like shown here, but there's also pumps here in the sieve tube element. So either way, it would be considered loaded. So once sucrose has been loaded into the phloem cells, what is going to happen to the water potential in the phloem cells? I want you to pause the video and think about that. Okay, hopefully you said that once the sucrose has been loaded in to the phloem cells, that additional solute that's been added in is going to lower the total water potential inside the phloem cells. This is going to be important when we move our sucrose from source to sink. Okay, so we have our mature leaf, right? And this is what made the sugars, and ultimately it got converted into sucrose, and we're going to load it from our source to our sink, which in this case is a pumpkin. And so at this point, we're saying that sucrose has already been loaded, right? So if it's been loaded, and so this group of cells right here, this long group of cells, those are the sieve tube members. And then this little tiny cell right here, that's the companion cell. And these would be the cells that either made the sucrose or one of the cells that kind of connects it to a more distant cell that made the sucrose, either way. And then this column of cells, those are the xylem cells, so they're transporting water. Okay, so here we go. We've now loaded our sucrose at the source, so consider sucrose to be loaded here. Once sucrose has been loaded into a sieve tube member, that decreases the total water potential in the sieve tube member. That means that the xylem tissue next to it has a higher water potential. This will cause water to be transported laterally from the xylem into the phloem cells. And, oh, sorry. Oops, one more. Again, this will cause water to be transported from the xylem tissue into our phloem cell, and that will increase the water that is now inside the sieve tube member. That's going to put pressure inside the cell. That pressure that's moving into the sieve tube member is called hydrostatic pressure. So we've now increased the hydrostatic pressure at the source. Once hydrostatic pressure is increased here, that means automatically pressure at the sink is lower. So therefore, the sucrose will be transported from high pressure to lower pressure near the sink. Once it's been moved to the sink, then it will be moved or it will be unloaded at the sink, right? So move towards the sink due to differences in pressure, and then we will unload it at the sink, so into our pumpkin cells in this example. But when sucrose has been unloaded, that increases the water potential in the sieve tube member, and that will cause water to then go back to the xylem cell, since the xylem will have a lower water potential. And then through capillary action, water will move back up. So one more time, here's our source cells that ultimately we have to get our sucrose loaded into our sieve tube member. Once sucrose has been loaded into the sieve tube member, we have now decreased the water potential due to the excess of solute. That is going to cause a higher water potential in the xylem tissue. Water then gets transported from the xylem into a nearby sieve tube member. That increases the pressure, hydrostatic pressure at the source. This difference in pressure will cause the phloem sap to be transported from the source towards the sink, which has a lower pressure environment. This less hydrostatic pressure is what allows for that movement from the source to the sink. This is where sucrose will then be unloaded into our sink cells. Once sucrose has been unloaded from the sieve tube elements, that causes an increase in total water potential here at the sink, and that's going to cause water to move back towards our xylem tissue, which would have a lower water potential, and then through capillary action, it will move back up. So it's kind of like water's moving up anyways because of root pressure and transpiration, but then some of the water will take a detour to help move the phloem sap by translocation. but ultimately that water goes back, right? Just took a quick detour. And now it's back in our xylem tissue. There's another picture. It just shows the same thing, right? So we loaded the sucrose here. Water then gets transported due to a lowered water potential, increase in hydrostatic pressure. The difference in hydrostatic pressure pushes or translocates the sucrose towards the sink, which has a lower pressure, and then we unload it at the sink. Once unloaded, that increases the water potential here and moves the water back over to the xylem tissue where there was a lowered water potential. And then the water just moves back up by transpiration. Most diagrams that you will see are almost always going to show translocation moving down. In fact, it's really hard to find a diagram where you see translocation of sucrose molecules moving up. But that doesn't mean it doesn't happen, right? We've talked about this. Sucrose can be transported in all directions, so you must always use source to sink. Here's like the only diagram I could actually find that does show the sink above the source. But in reality, it can be above the source at any time. It just depends where are we moving it to. If the flowers that we're moving it to are higher up and they're our sink, then we would be translocating in the upward direction. That's why you always just need to say from source to sink. Don't say up, down, left, right. None of that for sucrose transport. Source to sink is the magical phrase. Okay, I want you to pause the video and study this plant, and then I want you to see like if you can identify different sources and different sinks. Okay, hopefully you paused the video and you came up with mature leaves, so all these like really big leaves, those are going to be our sources. And then our sinks are going to be small, immature leaves, fruits, flowers, roots, and then also these root vegetables, the tubers down there. And then ultimately, these sinks can become a source later if the plant is on a life cycle that would pull from those stored carbohydrates. And you might have noticed that this is kind of an odd plant. It's like a tomato-potato plant. So it's not a real plant, but it is a fun plant to kind of talk about in these notes. Okay, and we did kind of touch on this in the part 2 notes, but I am going to review it now. That all of these cells that are transporting molecules, like our xylem cells, transport water, and then our phloem cells are transporting our sugars. Remember, they have adaptations that allow for them to have this type of function, which is transport. And so there's a lot of adaptations that our sieve tube members and companion cells will have to accommodate translocation of the phloem sap. So one thing we previously mentioned is the fact that the sieve tube members are hollowed out. This happens during development. As the sieve tube member matures and is going through differentiation, becoming a mature cell, pretty much everything from the cell membrane inward, which is all collectively called the protoplast, gets broken down and removed from that cell. And that allows for these cells to, even though they're alive, have more space for this bulk movement or mass movement of sucrose transport. We also said that each cell is connected end to end and that they are connected by sieve plates. So there are these openings between the sieve tube members to allow for sucrose to be transported from cell to cell to cell. We also said that our sieve tube members have really tough cellulose embedded within their cell walls, and this allows for them to withstand the high pressure environment that will occur during translocation. Remember, we have that hydrostatic pressure building up, and we need our cells to be strong enough to withstand that level of pressure, and they are. And then we said that even though the sieve tube members are alive, they really lack a lot of essential things that keep a cell alive. And so there's a companion cell that is going to be attached to it, adjacent to it right here, that has these plasmodesmata connections between them. And they're typically like large plasmodesmata, because what happens is the companion cell is going to constantly be feeding the sieve tube member whatever it needs. So proteins, ATP energy, carbohydrates, whatever it needs, it gets transported from the companion cell to the actual sieve tube member. And that's it for the part 4 notes. So please let me know if you have any questions.